All right, so now we're going to continue on with the exam objectives from the heart chapter instead. If you are enjoying my little decorations here in my office, if you come to my office for office hours or something, you may be able to see even more. So if that's a lure for you, that'll be good. Okay, so let's take a look at heart. So for the heart chapter, we really have two different exam objectives that we need to go through. One has to do with comparing and contrasting the action potentials that occur in the two types of cardiac cells, the contractile cells and the autorhythmic cells. And in essence, we're kind of comparing what we have learned previously in terms of skeletal muscle and its action potentials with what we're learning now with the heart, and even, skeletal muscle, but also neurons, right? So they have really fast action potentials on the order of like 6 milliseconds, and it has a very quick depolarization due to sodium, a very quick repolarization due to potassium, and a little bit of hyperpolarization before we go back to a resting potential. And it generally starts at about -70 millivolts. At about -55 millivolts is where we have the voltage gated channels, sodium channels opening up. And so we have that massive movement of sodium into the cell that depolarizes it. And then the potassium ions leaving the cell is going to repolarize it. Okay, so what do we get now when we're looking at the autorhythmic cells and our contractile cells? So this is a graph of the action potentials of what's going on in the membrane potential of our pacemaker or autorhythmic cells. So again, when we're doing these comparing and contrasting types of questions, you can write it as a paragraph in terms of just writing it out as an essay form of what's going on with these different types of cells in this particular case, or you can do comparisons that are in more of an outline form. That's totally fine as well. So it's cardiac contractile cells over here, autorhythmic cells here, comparing them in terms of resting potential or depolarization, repolarization, plateau, duration, things like that, right? So you can just list them and then how they are similar or different. So comparisons with things that we see here, we don't see a resting potential, right? There's no area of the curve that is flat. It's constantly depolarizing, then repolarizing again and again. We see that it starts at about -60 millivolts as opposed to -70 like we learned before. We see that the depolarization has two parts. We have a slow depolarization that is from sodium, and then we have a very fast depolarization due to calcium, okay? Now, the repolarization is the same as what we learned before. It's due to potassium ions moving across the membrane, and then we go back to it immediately again, okay? So we can compare our depolarization, our repolarization, our resting potential, or lack thereof, right? Things like that. All right. In addition, we need to know what cells are contributing or where these different cells are located, right? So these are autorhythmic cells, and these are going to be found within the conduction system of the heart, like the SA node, the AB node, Purkinje fibers, things like that. So then let's look at our contractile cells. So in fact, down here is what we see as the typical form for the action potential that's occurring in contractile cells. And I'll also shift to a graph that is 2 slides later, but notice the difference of it, right? We definitely see a resting potential, and we see it's very elongated, right? We're still in milliseconds here, but we see 200, 400, 600 milliseconds as opposed to what we were seeing before, right? In terms of like skeletal muscle and neurons and things like that, where it's on the order of 5 to 10 milliseconds. So we see the duration is quite a bit different with our contractile cells as well. It has a very long action potential as well because it has this plateau to it before it starts repolarizing. So let's look at the next curve that we see here. And so here's the action potential that we see here in blue. And then what they're showing you in purple is how the muscle then responds as the filaments slide, right? When we talk about the whole sliding filament, how it starts generating tension. But in terms of the electrical activity, this cursive R-shaped action potential is what we're talking about. And you can see that it's about 300 milliseconds, right? So much longer. We can see that it has a resting potential at about negative 90 millivolts. And then we have the depolarization, a protracted period of depolarization called the plateau, and then a repolarization. This depolarization comes from, again, sodium influx. And we see that over on the right side here in terms of permeability. We see sodium going into the cell, as we have seen in other cells, in order to depolarize it. But then this plateau is due to calcium channels, right? So the plateau that exists here, which is unique, is due to calcium going into the cell, which maintains it in a depolarized state before we then repolarize again with potassium exiting the cell. So potassium's the same in terms of its role. Sodium's kind of the same in terms of its role, but we have this unique plateau here, as well as an extended action potential with the plateau, and then an extended refractory period as well. Because remember that in this case, we want to make sure that the different chambers of the heart are really squeezing to eject the blood, and that's why we have this kind of longer contraction and longer action potential, and then also giving them a chance to relax and fully fill with blood before they contract again. Okay, so that is what we need in terms of our first one, where we're comparing the autorhythmic and the contractile cells with their action potentials. The next one that we need to go through is this one, right? So it's the ECG or the EKG, depending on where you're from. So in this case, we need to know the active waves and what's going on both electrically and mechanically. And when we look at this, remember that even though we're showing electrical activity in this graph, it's not of an individual cell like we saw in the other ones. This isn't an action potential. So going up isn't depolarization, coming down isn't depolarization. Each wave that we see there is like a burst of activity. It's kind of like a survey saying how many cells are really active in what's going on in the walls of the atria, and then the walls of the ventricle, etc. All right, so we have the three waves that you need to be able to identify: the P wave, the QRS wave, and the T wave. The P wave is atrial depolarization. So both atria, these cells are depolarizing in preparation for contraction. So the atrial cells depolarize, these contractile cells, and then they respond by contracting right afterwards, right? Afterwards, we see the QRS wave complex. This is both, and don't forget this, both ventricular depolarization, where they're starting to get the signal now, and atrial repolarization. They overlap during the QRS wave complex, okay? So the atria are going to go into their relaxation and filling stage as they repolarize. Meanwhile, the ventricles are going to go into their contraction phase as they get the depolarization signal, okay? So that's what's occurring during the QRS wave. And then finally, during the T wave, we have the ventricular repolarization. So they're going to repolarize and then go into their relaxation and filling stage. And then the whole sequence starts again. Okay? Okay. So that's what we needed to know for the QRS wave and the P wave and the T wave that make up our repeating electrocardiogram output. So that should do it for the heart. We will move on to doing the blood vessels on the next one.